She received her PhD from Germany and postdoctoral experience at Harvard Hughes Medical Institute, USA. Today, she'll be discussing with us about her research, and her talk is titled Drosophila melanogaster as a model organism to study brain development. So, we will go ahead with Dr. Sonal.
Hi. I will not turn on my video. I hope the presentation goes smooth with this hotspot this thing. So I'm going to talk about how we can use Drosophila melanogaster as a model system to study brain development. So as we all know that brain is the most important organ of a human body, and it consists of several parts like cerebellum, brainstem, cerebellum, and what it does is basically it controls most of the activities of our body. It collects information and processes, integrates, and then coordinates with other body parts to basically execute a function. So different parts of brain have different function that you can see here. Like cerebellum here is involved in basically balance, coordination, and cognitive learning. And brainstem helps in keeping the heartbeat and breathing properly. So a human baby is born with about 86 million neurons and many more neuroglia that support and protect the neurons. So if you want to see this in numbers, this is how it looks. So these many neurons and more neuroglia. And all these cells are basically born from a bunch of specialized cells that are called neuronal stem cells. So these neuronal stem cells, they basically arise during very early during gastrulation, about 2 weeks, and from a very specific layer, which is called ectoderm here. So a couple of cells from ectoderm, they start to uh, differentiate or develop into cells called neuroectodermal stem cells. So the major signaling pathway that is involved here is not signaling pathway that actually defines these cells. So once these cells are formed, then they will first divide to make a large number. Large means a couple of hundreds of these. And then they start undergoing specialized divisions that I'm going to explain in a bit. These are called asymmetric cell divisions. And then they form all these neurons and glial cells to basically form a complete brain. So now if we look at the timeline of brain development in human, it is like, as I said, that these like these neuronal stem cells, they delineate about 2 weeks, in about 2 weeks. And then at week 3 uh, 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 of pregnancy, the brain neural tubes start to form. And then by week 10, we have brain basic regions of brains are forming. And then about week 25, these neurons, different neurons start to connect to each other and form synapse. And this actually goes on until the full term, which is 40 weeks. And even after that, the synapse or the connections are form until late, very late in life, basically. So since we see it's a long time, 40 weeks, and if you want to understand how the brain developed, how these neuronal stem cells are born, how do they form neurons and glia, and how are these connections are formed, it's very difficult to study all these steps in humans. First, the time, and second, we will need a lot of brains to basically look at these processes, which is not possible. So what do? We actually turn to model organisms. So as Dr. Megha already told you about, there are several model organisms that we can use to study basic biology and also different disease pathogenesis. So here I'm going to talk to you about one specific model organism. It's a very small fly. It's called Drosophila melanogaster, and common name is fruit fly. I'm sure you all have seen these flies. Usually they come around rotten food or fruit, basically. So why do we choose this? So the reason is that this tiny fly has about 14,000 protein coding genes, and out of these genes, about 60% are conserved in human. So basic function of these, most of these genes are conserved in human and Drosophila. Moreover, the genes that are known to cause disease, like mutations in genes that are known to cause disease in human, so about 70% of them are also conserved in this tiny fly, which is called Drosophila. So we now use this model, Drosophila, to study basic biology of development and brain development, behavior like aggression, and regeneration like exon regeneration. And apart from this basic biology, we also use now these flies to model different diseases like, or to study different diseases like metabolic syndrome, aging. You can hear me, right? I'm still there. Hello?
Yeah, we can hear
Yeah,
you.
okay, okay. So metabolic syndromes, to study aging related stuff, cancers, and also neurodegeneration. So when we look at the life cycle of Drosophila, it's about only 10 days, which starts from a small embryo, and then it goes through three different larval stages: first instar, second instar, and third instar. So during this time, this larvae basically they just keep on eating food and accumulate, so that they when they undergo pupal stage, when they cannot eat, they develop all the organs, important stuff. And then after pupal stage, we have adult stage, and then the cycle goes on basically. So if we compare the timeline of brain development with human to Drosophila. So in human, as I said, that it takes almost 40 weeks to develop the basic brain structure. In Drosophila, it takes only 10 to 12 days basically. So in Drosophila also, the early neuronal stem cells, they arise from neuroectoderm during embryogenesis, which is about 10 to 12 hours after egg lay. And then we have these neuronal stem cells. They start to divide. They divide and uh, attain a proper number, which is 200 of total of the stem cells in Drosophila. So they first divide and get populated, and then they start dividing again to give rise to all the neurons and glia. So what happens between the embryonic stage and larval stage? Once the number is achieved, and then there is enough number of neurons for larva, these neuronal stem cell undergo quiescence. It's a dormant phase. It's a G0 or G2 stage where these neuronal stem cells they become small and they do not divide. And once the larva hatches, it finds food around, and once it eats food and there is basically free amino acid in the body, then these stem cells get a signal through different signaling pathways, and then they exit this phase quiescence, and they enter into mitosis and they start dividing, and then they keep on dividing for this first instar, second instar, and third instar phase. And during this time they make almost 99% neurons and glial cells that an adult fly needs basically. And the entire thing goes for about 10 days. And again, at once the pupal stage comes, some of these neuroblasts differentiate permanently, so they will never divide again. But some of these undergo the dormant stage or to Z0 or G2, which is basically a quiescent phase, which then are they are sit in the adults until they need to divide again. So I will discuss a little bit about it uh, later also. So now we know it takes 10 to 12 days while we compare with human. So how does it look like? So you see here, this is Drosophila embryo, and this is the nervous system of an embryo where we have about 200 neuronal stem cells. Then they divide and give rise to all the larval neurons during the three stages. And this is how a larval brain looks like. The structure that you see here is called learning center, which is there in adult brain also. It is equivalent to human hippocampal region. And then this is the adult brain where we have about 250,000 neurons. No, there should be one more zero. 250,000 neurons here. So we see that in 10 days we attend all these neurons. So how come these small bunch of cells, which are called neural stem cell, give rise to so many cells? So as I said, they undergo a special division, which is called asymmetric cell division. So during asymmetric cell division, this neuronal stem cell divides unequally. So normally during symmetric cell division, A cell divides into two equal daughter cells, and then they again divide. Here, what happens? This cell divides into two, give rise to a big cell and a small cell. The big cell becomes stem cell again, and the small cell becomes either intermediate progenitor cell or another cell type which is called ganglion mother cell. This then divides symmetrically and give rise to either two neurons or two glial cells. So what controls this asymmetric cell division? So we have, I'm explaining it here. So there are a bunch of proteins. So this is again in Drosophila so far. There are some proteins that localize at apical side here. These are called apical polarity protein. They are involved in many different functions. So as Dr. Mega said, you need spindles. They should be oriented properly. There should be a difference in sizes. So this complex spins mud and GL5. This complex controls the localization of proper like spindle point. And these other proteins actually define the plane where should be the um, cleavage furrow for cytokinesis. And then we at the basal side we have these proteins Miranda and Prospero and Brad. These are called so especially Pros and Brad, they are called cell-fate determinants. So what is cell-fate determinants? These proteins decide that the basal cell after division should start expressing genes that will later give rise to neurons or glia. So now once this cell divides, here is the plane of division. So it becomes a big cell. This becomes a small cell. This neuronal stem cell retain or actually get all the polarity protein here. So it still remains as neuronal stem cell. While the small one doesn't get these proteins, it gets the cell-fate determinants. So that then later it divides symmetrically and it gives rise to neurons or glia. And now this neuronal stem cell grows again in size and then the cycle goes on. So how does it look in Drosophila brain? So here, if you look, this cell is getting ready for division. All the chromosomes are coming at the metaphase plate with the spindle. Then the chromosomes are split into two sides. You can see the blue protein, which is the apical protein, which you can see here, and the green protein, which is self-fate determinant. You can see it here also. And then these are green are spindles. So now it is dividing. So this divides here as a big cell, which will again become the neuronal stem cell. And this is a small cell, which will be a GMC or ganglion mother cell. And this is the next step. You have a big cell that is going again for another round of division, the small cell that divides, and you see these are all neural cells or glial cells here. So this is how it looks like in a Drosophila brain. So what is, as I said, a lot of these proteins are conserved. So most of these proteins were first identified in Drosophila brain, and then later they were found in other model systems like mice or also in human. And most of these were actually implicated in some diseases, mostly cancers. So misregulation of these proteins in human lead to some type of cancer. Okay, so now we know that, okay, these neuronal stem cells divide asymmetrically and they maintain their pool of stem cells and also give rise to neurons. But then how do we, so at this point, how do we know? So there are so many different types of neurons. So how do these cells know when to make which neuron? They could all be same neurons, but it is also controlled in a spatial and temporal way. Means there is a series of protein, specialized protein, which are called transcription factors. So these transcription factors are expressed in these neuronal stem cell, but not at once. They're expressed sequentially. So one will come, the second will come after that, and third. And this is how they give identity to all these neurons or ganglion mother cells. So like here, if we look, the first one is hunchback. So this neuronal stem cell at very beginning, it'll start expressing this transcription factor. This transcription factor is passed and then it has different targets. So they are expressed in these neurons. So these are basically early born neurons. Then the same neuroblast after a neuronal stem cell, after some time, it will start expressing another transcription factor. So the same neuronal stem cell, now after some time, it is expressing another transcription factor. Then this transcription factor is passed to this because it is a sulfate determinant. So it is at the basal side. Basal side, then the ganglion mother cell is getting it. So now it is expressing another protein. Then this protein will be transferred to glia or neuron cell. Then again, the third one and fourth one. So this way we see basically a series of different neurons. So these neurons are different than this and then this and then this. And it is also possible that some of these neurons express two of these. So then that's how we get the diversity. So over time during development from embryonic stage till larval till adult stage, these different transcription factors are expressed and they give rise to different types of neurons. Then they go or they migrate to and reach different parts of brains where they form different they form connections with different types of neurons, and the entire brain is formed while the development is still going on. Okay. So now we know that there are different cell types or different neuronal stem cells that give rise to different progeny. So how do we basically um, see what happens in adults? This is all during development. So a stem cell is dividing asymmetrically, giving rise to different neurons. They migrate and then they form the entire brain during development. What happens once the development is complete? So during so once the development is complete, some of these neuronal stem cells, they are kept still alive and in a dormant stage or quiescent stage. These neuronal stem cells, they stay like this until there is a new memory or there is a new thing that brain is learning, or mostly until there is an injury. So they are in quiescence, they are not dividing. But if there is an injury, these stem cells get basically activated and they start to make the neurons, and depending on where their location is, they will make the progeny for that area or neurons of that specific area. However, this process is slow, it's not very fast, so it depends on the the degree of injury, and based on that the human, sometimes we can regenerate and sometimes there is no way we can regenerate these neurons basically. All right. So now so what do we do with it? Why do we So these processes are highly regulated. Why do we have to study them? So think about it. When things are going properly, we have a complete brain which is normal. What if these neuronal stem cell, they decide that we do not want to self renew, or we do not want to undergo these asymmetric division? I just want to divide symmetrically and just want to give rise to two neurons. So that happens in some cases where the polarity protein are basically mutated, or polarity genes are mutated. Then we don't have those proteins, and then in that case sometimes it happens that these neuronal stem cell, they basically divide asymmetrically, or and prematurely differentiate and give rise to neurons and glia. In this case we won't attend the number of neurons that we should attend, so it will lead to neurodevelopmental disorders. And there are several, and one of the one that Dr. Megha also explained is microcephaly. So microcephaly is basically a result in most cases where the neurons either die, or they undergo premature differentiation, or they just stop dividing. So microcephaly is basically a condition where a baby is born with a very small brain size compared to the normal kid. And there are 2 to 20 cases about in 100,000 births, and in India it's about 2 per 10,000 births. And depending on the cause and severity, the kids can also have very different problems like intellectual disability, developmental delay, epilepsy, and also several other like problems related to ophthalmology or hearing disorders. And it's a condition which is a lifelong condition, and so far there is no cure for this. So how does a kid look like? As she already showed, that the kid who is born with a very small brain, they they have microcephaly basically. So what happens when the neuronal stem cells, they decide so that we should keep self renewing, They do not want to go under asymmetric division. They just want to divide symmetrically, means they are giving rise to neuroblast or neuronal stem cell all the time. Or at the same time, what happens if these stem cells that are quiescent in adult, they suddenly exit, one of the protein which is important for maintenance of the stage, quiescent stage, is gone, and they randomly start dividing. This all lead to, or another situation is when these cells, when they should ideally I should ideally differentiate, but actually they de-differentiate back, means they gain the property of stem cell and they start to divide asymmetrically again. This all lead to basically brain tumor or cancer. So um, uh, so to understand all these things, as I said, that we have a nice system that I'm using in my lab right now as a Drosophila melanogaster. So what people can do with it? So I'll show you one example which is not from my lab, but it's a very beautiful example. So now we have a model system. We know these proteins that are involved in asymmetric divisions or differentiation or quiescence, they're all conserved and they play the same role in Drosophila as they play in human, which people are now finding out. So how do we use this Drosophila to study a human disease, or to define or to detect whether how then diseases being diseases arised because of loss of one gene. So what has it's for the gene is called ankle 2 in Drosophila. So in my postdoc lab, basically what they did, they found they we did a large screen in Drosophila, basically using mutants, generated a lot of mutants, and we wanted to find out whether these genes they have any role in human, or they are conserved and their function is conserved, and whether they lead to any disease in human. So here there is this family, and this family had two kids. Both have microcephaly. The girl died when she was 3.5 years old, while the son was almost about 5 years. But he had severe problems. He had strong microcephaly. You can see the brain is small. So this kid is 5 year old, but he looked like the 1.5 year old kid, and he had a muscle problem, and he couldn't eat or feed by himself, so he would just sit. And here you can see his brain, which the brain is very small. There are very few gyri basically here. So when this family came to this hospital where we were, and then they said, okay, the kid has this problem, what can we do? Can we find out, because the first daughter was also the same, so can we find out which gene is causing this disease, and is there a way to cure it? So then what the group did, they looked at the potential gene in this kid where there were some mutations. So they pulled out, there were 5 genes, they had mutations, but they didn't know which one of these 5 gene could lead to this disease. So then they looked at these 5 genes, and to find out whether Drosophila has these 5 or not. So in Drosophila, all these 5 genes are conserved. And then they looked, so we had generated already mutants. So we finally looked which mutant or for which gene leads to the same phenotype, which is microcephaly phenotype. So we figured out that there is this gene ANKL2. Loss of this gene actually leads to small brain. So here what I am showing you is Drosophila larval brain. This is the normal brain in control, and this is this gene mutation in this gene lead to small brain. So now, okay, we see out of 5, this gene mutation in this gene Drosophila leads to the same phenotype as human. But is it really the gene, whether they are they have the functional conservation, whether the gene has the same function in human and Drosophila? So for that, what it basically did, they took that human gene sequence from human, and they introduced in Drosophila. So Drosophila has no protein from its own gene, but now it gets a copy of human gene. So basically in Drosophila, the human gene is protein is being made. So this human protein actually rescued the phenotype, means it actually rescued the problem of small brain in Drosophila. So this act in the wild type condition, when they went back and took the mutant, the mutation that causes this, and then they express the mutated gene protein in Drosophila, that did not rescue the phenotype. So here it confirms that this is the gene which is causing small brain phenotype or problem in this kid. So then the next step was to basically go for IVF, in vitro fertilization, look for embryos that do not have this mutation, and then they selected one of that, and now they have a healthy kid. So this is how we can use this model system or any model system to study disease and to find information about, disease causing genes. We can also look for the biology. So later the same gene was associated with Zika, where they found that in Zika virus, this protein, so Zika virus infection, during Zika virus infection, somehow the mRNA of this gene was downregulated, and that lead to basically the small, one of the reason that Zika, in Zika patient they have small brain basically. So overall I can actually with this conclude that we have a nice system, model system, that we can use to study brain development in uh, Drosophila, because most of these genes that are involved in uh, neuronal stem cell development, maintenance, and differentiation are actually conserved in human. All right. Thank you.
Okay. That was fantastic. I really liked how you started off with, some of the basic aspects. Let me just go to the views. Okay. So I think the entire process of neuronal development that you discussed was very interesting. It was kind of very refreshing for all of us and very new to the students, I think. And I think the case study actually was very good because it put the entire thing into perspective as to how you are actually able to relate the work that you're doing in lab to the people or the common people. So thank you so much for that discussion. We have a couple of questions here. I'll start off with some basic questions and then something very interesting. So one of the participants asked, I think in the very first one or two slides, you used the word myelination.
Uh-huh.
I think they wanted to know what myelination means.
Okay, so myelination is basically a sheet that covers the neurons basically, so to protect it from different injuries. In Drosophila, we do not have myelination, but in humans and other systems, you have a sheath which is of myelin. It's a rich, substance that actually surrounds all the neuron axons to protect them or to insulate them. Because at the end there is a firing, and you want to protect them. So this is a sheet of myelin around all the axons,
Okay.
which we do not have in Drosophila.
Okay, sure. So the next question is, I think, a imaginative question. What happens if our left hemisphere is larger than the right hemisphere in the brain?
What happens when it is so actually what I to be honest, I won't be able to say it correctly perhaps, because I am not working at human this thing, but actually it is what I know is that when you use one side of body, like then the other side is connected, and that is working. So I one side is big. Anything which is bigger than normal, of course it is not good. But normally there is saying that if you use your left hand more often, then your right brain is more active. I'm sorry, I cannot answer that correctly.
I'm sure that probably is not your specialization. So,
Yes.
yeah, yeah. So, another question related to the case study that you discussed was
Mm-hmm.
amongst twins, does
Mm-hmm.
microcephaly happen in both or only one of the babies?
So it depends on, basically it depends. So if you have a mutation that is coming, so there is always a 50% probability, depending on if mom is carrier, or the dad is carrier, or if it is coming from which like depends on which chromosome. So in this case, in Drosophila, It's on X chromosome. Okay.
Oh,
So what we had, and if we are taking male, and all the males have this mutation, so basically all the babies will have this, because it's mostly, or all the female babies will have
female
this. But
babies.
in twins, it again depends. In this case, these people have didn't have twins, but from the fertilized embryos, there were a couple where they had this combination, where the moms mutated allele, and the dads came together, and then the baby had disease. But when both the normal allele came, and then there was no disease. So it depends.
I think one last question maybe. So, if people have severe small brain disease, or I think they are trying to talk about the microcephaly,
Microcephaly.
does microcephaly eventually lead to death, or do they
Yes.
survive?
Yes. So it does lead to basically death, basically, because most of the functions, I mean, you are not generating enough neurons, so
Hmm.
you won't be able to eat, or you won't be able to walk, or you won't be able to think. So you can be dependent for some time, but there are many basic functions that where you need, like breathing. So eventually, one or the other things fail, and then it mostly it leads to death.
Okay. So I think the questions have stopped coming up. So it looks like this was a very engaging talk, and people really understood most of it. So I think this brings us to the end of the morning sessions for today. And we will resume after the lunch at 2 o'clock, where we will have a talk by Dr. Pavitra Chavli, where she'll be talking about clues to health and disease on many organs outside the body. So I would like to thank all the panelists since morning, and all the speakers of today, and more importantly, all the audience for being such engaging audience, and inspired to ask good questions, learn from all the scientists out here. So thank you so much. So we will resume